Hello, friends. Jim here. Interesting article that appeared in the online publication phys.org. Polar experiments reveal a seasonal cycle in Antarctic sea ice algae. Well, of course, there's seasonal changes, so you would expect that. What's really interesting is the mechanism. So here we have a photo of microbes turn the underside of the sea ice brown or green, sometimes forming thick mats in the, on the underside there. In the frigid waters surrounding Antarctica, an unusual seasonal cycle occurs. During winter from March to October, the sun barely rises, depends on your latitude. As seawater freezes, it rejects salts, right? Brine rejection. You've heard me discuss that mechanism numerous times. These create pockets of extra salty brine where microbes live in winter. In summer, the sea ice melts due to constant daylight, producing a warmer situation, but by melting the ice, you get a freshwater lens at the surface. So the Southern Ocean, right? This ecosystem is home to much of the Southern Ocean's photosynthetic life. A new University of Washington study provides the first measurements of how sea ice algae and other single-cell life adjust to these seasonal rhythms, offering clues as to what might happen as the environment shifts due to climate change. The study was published in the ISME journal, and it contains some of the first measurements of how sea ice microbes respond to changing conditions. We know very little about how sea ice microbes respond to changes in salinity and temperature, said lead author Hannah Dawson, who is a University of Washington, UW, postdoctoral researcher who did the work while pursuing her doctorate in oceanography at UW. And until now, we knew almost nothing about the molecules they produce and use in chemical reactions to stay alive, which kind of important because, if they stay alive, then they can support higher organisms. But here's the other interesting thing, all right? So how do they stay alive due to what's happening to climate impact, and then what does it mean for carbon storage? And this is the interesting bit, cloud formation. Stay tuned for that. Polar oceans play an important role in global ocean currents, right? Deep water formation, the upwelling, right? And so on. You've heard me discuss these numerous times before. And supporting marine ecosystems. Microbes form the base of the food web, supporting larger life forms. Microbes can be phytoplankton, it can be nanoplankton, which is bacteria, and that's the focus of this research here. Polar oceans make up a significant portion of the world's oceans, and these are very productive waters. Southern oceans actually contain some of the most productive open water productivity on the planet, right? Let me repeat that again. The productivity in the Southern Ocean is the highest anywhere in the open ocean. Very important region. And that statement was from senior author Jody Young, who is a UW assistant professor of oceanography. These waters support big swarms of krill, the baleen whales, right? Penguins, orcas, right? So on. At the start of the ecosystem are these single-celled microscopic algae, and we know very little about them. The tiny organisms are also important for the climate since they quietly perform photosynthesis, removing carbon from the atmosphere. Polar algae are especially good at producing, this is the kicker, sulfur-containing molecules. They give beaches a distinctive smell, but when lofted into the air in sea spray, promote formation of clouds that can reduce penetration of solar rays. In other words, the more clouds, the less direct sunlight, the less UV energy, the less heating of the waters that can take place. But how does that factor into the overall Earth energy imbalance? You hear a lot about that recently. I still contend that by not looking and considering the contributions, the various aspects of the contributions from the ocean, the EEI is not providing a complete picture. In other oceans, satellite instruments can capture dramatic seasonal phytoplankton blooms from space that isn't possible for microbes hidden under sea ice. And Antarctic waters are particularly challenging to visit. That's an understatement. Basically, they often do not get measurements during the winter. So in late 2018, Dawson and co-author Susan Rundle traveled to Palma Station, which is a U.S. research station in the West Antarctic Peninsula. They got into a small boat, sampled seawater and sea ice at the same nearby sites every 3 days. At the time, she was a graduate student. In fact, they both were. So they performed 10-day experiments in tanks to see which microbes grew as temperature and salinity were adjusted to mimic sea ice formation and melt. They sent samples back to Seattle for more complex measurement of the sample's genetics and metabolites, basically organic molecules the cells produce. Results revealed how single-celled algae deal with their fluctuating environments. As temperatures drop, the cells produce cryoprotectants, basically antifreeze, right? Cryo, right? Protecting from the ice, to prevent their cellular fluid from crystallizing. Many of the most common cryoprotectant molecules were the same across different microbial life forms. And here's a photo of oceanographers at work. Right? You collect your samples into here. And this is the photo of the experimental setup. Right? So this is how we do it, folks. Sometimes you get in these little boats. Not always the big research vessels. As salinity changes to avoid either bursting and freshening waters becoming desiccated, the cells change the concentration of salt-like organic molecules. Many such molecules serve a dual role as cryoprotectants to balance conditions inside and outside the cell to maintain water balance. Okay, so let's kind of take a step back here to look at this a little further. There's a tendency that if you have, a fluid with a salt concentration, and let's say there is a perforated barrier separating fluid A from fluid B, and both of these fluids have a certain level of salts in it, but they're different. There is a tendency for water to flow from one side to the other to try and balance out the concentration so that you have the same concentration of the salts. So, for example, if fluid A has a much higher salt level than fluid B, then you're going to have water from fluid B flow into fluid A to dilute. Therefore, now let's and vice versa. So fluid B has too low a salt concentration, so it wants to get rid of the excess water in an attempt to get to an isoosmotic or osmotic balance. Consider this a little further. Let's start with a freshwater fish. If you look at the concentration of salts in a freshwater fish's tissues relative to its environment, it's much higher in the fish. So there's a tendency for water to flow into the fish's tissue. That has the, if it's not regulated, that has the danger of bursting the cells. Right? So the fish would be considered hyperosmotic relative to the environment, where the environment is hypoosmotic relative to the fish. So the fish, in being hyperosmotic to the environment, has a tendency to draw in water to try and dilute the salts. So the fish counters this by drinking very, very little water, but urinating quite a bit. And the urine is quite dilute. And there are special cells in the gills called chloride cells that help with this, as well as what the role of the kidney in the fish. Now, on the other side, a marine fish, its salt level is much lower. Its salt concentration is much lower than the environment. So it is hypoosmotic relative to the environment. So there's a tendency to increase the concentration of salt in the fish by pulling water out. So the marine fish actually drinks a lot of water, but the urine output is considerably reduced and quite concentrated. So basically that's what they're getting at here, is that at first, with all that fresh water, the microbe is hyperosmotic to the environment, so the water could flow in and bust it. But then on other situations where you have the major, the sea ice forms, you have brine rejection, so the salinity is increasing, then you have the flip side of it. So now they become hypoosmotic, so they're in danger of losing the fluid and shriveling, so they have to counteract that. And by the way, with the fish scenario I was describing, estuarine fishes, which live in a wide range of salinity fluctuations, they have to have the ability to do both mechanisms. Get rid of excess fluid and conserve fluids. They have to, depending where they're at, depending in the tidal cycle and so on, they either have to conserve fluid and excrete very concentrated urine, or excrete out a lot of urine that is very, very dilute. You have to get rid of the excess water. Save water, get rid of excess water. That's what the challenge estuarine fish have to do. Which is why when a salmon is moving downriver, right, it's living in fresh water. So it's employing the physiological responses I mentioned earlier. Copious dilute urine. Well, then it starts to encounter, it gets near the ocean, right? It's going into brackish water, whatever. There's a period called smoltification. This is when the salmon undergoes physiological changes so that it can now handle the conditions of the ocean. So now it has to change its physiology to basically excrete very concentrated urine, save as much fluid as possible, not have copious dilute urine. That's why it takes them some time before they make the transition, which is why on their return to spawn, part of the reason why they die is because their physiology is geared to dealing with the salt concentrations in a marine environment, not a freshwater environment. So we see similar things here at the microbe level. So the results show that under short-term temperature and salinity changes, community structure in each sample remains stable while adjusting the production of protective molecules. Different microbe species showed consistent responses to changing conditions. This should simplify modeling future responses to climate change. Results also hint that the production of omega-3 fatty acids may decline in lower salinity environments. This would be bad news for consumers of krill oil supplements and for marine ecosystem that relies on those algae-derived nutrients. So the research is investigating this further. So Dawson states we're interested in how these sea ice algae contend with changes in temperature, salinity, and light under normal conditions, then toss in climate change and what's their response to all of that. And of course, the climate change is just changing things, remodeling the landscape in terms of when sea ice is forming, how much sea ice forms, how long it stays before it melts, as well as the quantity of freshwater input from glaciers. So we're both trying to capture what's happening now and what's happening in the future. And don't forget, this past year saw the lowest level of Antarctic sea ice that we have data for. So that's really going to throw a wrench or three into the workings. And by the way, I did a video some time ago called Ice Algae. I talk about the productivity on the underside of the sea ice and so on and so forth. So you may want to check that out as well. So this is interesting. I would have liked to have seen more comments directed towards the sulfate aspect that was mentioned, because that sounds really, really interesting. So, but anyway, that was kind of an interesting point there about that they can create, that they can release sulfates, and this will impact solar rays penetration, sulfur-containing molecules. So this implication, I think, needs further examination right here, right? Polar algae is especially good at producing sulfur-containing molecules that, when goes into the air as sea spray, promotes formation of clouds that can reduce penetration of solar rays. This needs further investigation, and its role in the climate, AME, right, aerosol masking effect, EEI, etc. So that's something I'd like to see take place. But just to show you how nature always comes up with adaptations to things, cryoprotectants, so that the changing osmotic conditions do not kill the microbes. So thank you for your time.